composed of three main components. Let me sharing my screen. So DNA first is a polymer molecule which is composed by monomer called nucleotide. Okay. Nucleotide is composed by three main components. First is a bicarbon super. So in case of DNA is deoxyribose. Deoxyribose. Case DNA and ribose in case of RNA. The first phase group and a base nitric. Okay. So this is a structure of nucleotide. So first we have an oxygen, we have five carbon sugar here. So the carbon number one is the carbon right in the right of the oxygen. Here, carbon number two next to it, carbon number 3, 4, and 5. Okay, 5 carbon super. So next is a full phase group, which is attached into the carbon number 5. This is the phosphate group. This is the phosphate group. Carbon super. And the base nitric. The base nitric is attached to the carbon number 1. I put it as red, for example. Phosphate group number 5. and base nitric attached to carbon number 1. So this is 3 main components of a nucleotide. Okay. Here. So in a neutral pH environment, a nucleotide will have a negative charge due to the phase group. And we can see that here, the differences between the ribose and the deoxyribose is only in the carbon number 2. In the ribose is the hydrogen group, and in the deoxyribose is the hydrogen group. So here, take another color. Here, attack is the OH or the hydroxyl group. In case of DNA, in case of RNA or ribose sugar, okay, and it is the hydrogen atom. In case of DNA, DNA is stand for deoxyribose. Nucleic. So deoxy that means lack of oxygen. Okay. So one nucleotide combined with another nucleotide to form a dimer, the link, the phosphodiester link, which also is the covalent link, will catch from the hydrogen group of carbon number 3 of the first nucleotide to the hydrogen group in the phosphate group of the carbon number 5 of the next nucleotide. And that's why the direction from the top to the bottom equal the 5' to 3' end. Because in here, the carbon number 5 comes first and carbon number 3 come later. Okay? Yep. So, for example, in here, if you finish this, I will erase that. Okay. So I will erase all of this. So if this nucleotide is combined with another nucleotide in here, okay, we will have the phosphodiester link between the carbon number 3 and the phosphate group. So the phosphodiester link, or this is also called this is also the covalent link, which is really stable. Covalent link. So the direction from the top to the bottom in here is, we can see here the carbon number 5 coming first and the carbon number 3 coming later. So in here we can call this is the 5' end, and Why is my arrow is like this? So this is my 5 prime and this is the 3 prime end. And the direction from the top to the bottom is called the 5 prime to 3 prime direction. Okay. This direction is really important. Sorry, I have a phone call. I will come back. Okay, so like you can see in here, the 5 prime to 3 prime direction. And in DNA, we have 4 types of nucleotide. So in here, the two first part, I say these two parts. These two parts, a 5 carbon sugar and a phosphate group, is identical. So these two parts are identical between nucleotide. Is nothing different of each nucleotide with these 3 parts. Only the base matrix is different. This part is different. So there are 4 types base matrix. A is stand for adenine. T is G is stand for guanine. C stand for cytosine, and T is stand for thymine. And this is also equivalent to 4 types of nucleotide, which is A, C, G, and T. Okay. So in RNA, we will have, instead of thymine, we will have uracil. And the structure of all the nucleotides was shown in the slide. So with the A and G is two big base matrix. We have two circles here. We call it purines. Okay, and with the T and C and U is 3 smaller one base matrix. We call it primitives. And you can see here the structure of thymine and uracil is quite similar. The only difference is in the position of carbon number 1 of the methane group is attached to the position 1 instead of the hydrogen bond in the uracil. That is the only differences between T and U. Okay. And we have a rule, complementary rule, or we call it Chargat rule because its name follows the scientists who discover this theory. That A and T is always linked with each other in 2 hydrogen bonds, and G and C is always linked with each other by 3 hydrogen bonds. So this is the position of 2 hydrogen bonds in A and T. This is position of 3 hydrogen bond between G and C. And normally the GC is have more 1 hydrogen bond than the pair of AT. So if a fragment of DNA contain more GC or consider a rich GC than the other, normally it will be more stable and need more energy, more temperature to break down and denature those DNA fragments than the others, because it contains more hydrogen bond. So it requires more energy, more temperature to break down and separate two DNA strands. So, for example, if in future you have to design to denaturate DNA fragments that contain many GC, so pay attention on that, because normally it will be much more stable than the DNA fragment contain of rich AT. One more thing is a complementary strand. So from this, are you finished with this? I will erase this. Okay. So if we have a double strand DNA, so this is a double strand DNA. Fragment. And I will zoom in one fragment. We will have, so the first fragment here, we have the nucleotide. And this thing? The ugly stuff. So let's take against that example that this is a DNA strand. We have 2 nucleotides and we have the direction from the top to the bottom here is 5 prime to 3 prime, right? This is the 5 prime. And this is the 3 prime direction. So now if we have a complementary strand here, we will have okay, it'll be like this. Okay, so position it upside down. I mark it again. So this is carbon number 1, which is in the right side of the oxygen atom. Carbon number 2, 3, 4, and 5. Okay. So now we will have another. Okay, so let's suppose this is the pair A adenine. So because link with A is T, so this it must be a T. Okay. Two hydrogen bond. A is linked with T is by two hydrogen bond. And this is the G and C. By three hydrogen bond. So as a result, if we have the direction from here, if 5' to 3', because the 5 carbon come first and the 3 carbon come later. So in here, we will have the 3 carbon come first and the 5 carbon come later, right? So as a consequence, we will have this direction from the up to the bottom is the direction from 3' to 5'. Okay, so in the double strand DNA, if we have the direction of one strand is 5' to 3', then the other, the complementary strand, will have the direction is 3' to 5'. Identify the direction of the DNA strand is really important because later when we discussed about DNA replication and also RNA transcription, we will see all the process, both DNA replication and RNA transcription is only happen from the 5' to 3', not the way around, not the direction around. So this is why we call this is an anti-parallel strand, because if one strand is from 3', 5' to 3', that means the other will come from 3' to 5'. So this is an example from of DNA double strand. So if we have one DNA strand, 5', 3' here is T, G, G, CC. Follow the Chargaff rule, the complementary strand will be 3', 5' of A is linked to T, G is linked to C, C is linked to G, and this G is linked to CC. And if we rewrite this negative strand from the 5' to 2', we will have to reverse it from CTA, so you can see CTA, GGG, DGA, and so on. Okay. Okay. Want to take a picture of this? You want to take a picture of this? Okay, take it.
You're not sharing the screen.
Professor, you stop.
Sorry?
You're not sharing the screen.
Sorry. Okay. Okay. All right. You finished? Okay. So now we will have a small exercise. Okay. Now we have a scientist is design a primer, which is DNA sequence or called a short nucleotide to use in DNA sequencing, and the DNA fragment most complementary with the template shown here. So in here is a DNA template which is provide from 3' to 5' with the sequence here. Could you spend a few minutes to provide me what is the sequence of the primer that complement with this DNA fragment in 5' to 3', please? So for easier for you, I will provide the sequence in the chat box, okay? Okay, great. Can you, when you send the solution, can you send to everyone, please? So everyone can, you know. Okay, thank you very much. The solution is correct. So for the solution here, we have to follow the Chargaff rule. A is always linked to T by 2 hydrogen bond, and G is always linked to C. And because the second is provided already in 3' to 5', so we just write it in here from 5' to 3'. So T is instead of A, C is instead of G, G is instead of C, A is instead of T, TA, and so on. Okay. So before going to the differences in DNA structure and RNA structure, let's watch a small animation on how the DNA works. What is the DNA structure?
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which 2 strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has 3 components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT or TA and GC or CG base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair nonspecific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule with us.
Okay, so next, I guess that we will have a small break, 5 minutes, before we go to the differences in the structure of DNA and RNA. Okay, 5-minute break.